All right, everybody. Hope you're doing well. Welcome to another Fix Your Gut live stream. I'm your host, John Brisson. Here is the Fix Your Gut website. If you haven't already, put in your email address and download a copy of the Optimal Health Blueprint, which are supplement recommendations for both males and females. If you need gut health coaching or general health coaching, or offer many different types of coaching with my vast medical knowledge, hit me up. I can get health and fitness coaching with my partner Jason Hooper. Pick up a copy of the third edition of Fix Your Gut. If you're having detoxification issues, I want to pick up some liposomal glutathione from Frontline Performance, which is the old bulletproof glutathione, the only one we recommend. Good probiotic that's even seems like safe to take for people who have histamine issues or delactate issues. Could try Smidge. Recent posts are here. I wrote a post last week on C-sections and their effect on infant gut health. Of course, the blog tab has been replaced by research. You can just click on it and you can see blog articles that we've written. All right, let's begin. New discovery explains anti-hypertensive properties of green tea and black tea. A new study from the University of California, Irvine shows that compounds in both green and black tea relax blood vessels by activating ion channel proteins in the blood vessel wall. This discovery helps explain the anti-hypertensive properties of tea and can lead to the design of new blood pressure-lowering medications. Published in the Cellular Physiology and Biochemistry, the discovery was made by the laboratory by Jeffrey Abbott, PhD, professor of the Department of Physiology and Biophysics at the UCI School of Medicine. Caitlin Redford, a graduate student in the Abbott lab, was the first author of the study titled KCNQ5 Potassium Channel Activation Underlies Vasodilation by Tea. Research results from the research revealed that two catechin-type flavonoid compounds, epicatechin gallate and epigallocatechin 3 gallate. You can get that from EGCG, from green tea extract supplements. Found in tea, each activate a specific type of ion channel protein named KCNQ5, which allows potassium ions to diffuse out of cells to reduce cellular excitability. As KCNQ5 is found in the smooth muscle that lines blood vessels, its activation by T catechins was predicted to relax blood vessels, a prediction confirmed by collaborators at the University of Copenhagen. We found by using computer modeling and mutagenesis studies that specific cations bind to the foot of the voltage sensor, which is part of the KCNQ5 that allows the channel to open up in response to cellular excitation. The binding allows the channel to open up much more easily and earlier in the cellular excitation process, explained Abbott. Because as many as one-third of the world's adult population have hypertension, and this condition is considered to be number one modifiable risk factor for global cardiovascular disease and premature mortality, new approaches to treating hypertension have enormous potential to improve global public health. Prior studies demonstrated that consumption of green or black tea can reduce blood pressure by a small but consistent amount, and catechins were previously found to contribute to this property. Identification of KCNQ5 as a novel target for hypertensive properties of tea catechins may facilitate medical chemistry optimization for improved potency or efficacy. In addition to its role of controlling vascular tone, KCNQ5 is expressed in various parts of the brain where it regulates electrical activity and signaling between neurons. Pathogenic KCNQ5 gene variants exist that impair its channel function, and doing so cause epileptic encephalopathy, a developmental disorder that is severely debilitating. It causes frequent seizures. Because catechins can cross the blood-brain barrier, discovery of their ability to activate KCNQ5 may suggest a future mechanism to fix broken KCNQ5 channels to ameliorate brain excitability disorders stemming from their dysfunction. Tea has been produced and consumed for more than 4,000 years, and upwards of 2 billion cups of tea are currently drank each day worldwide, second only to water in terms of volume consumed by people globally. The three commonly consumed caffeinated teas, green, oolong, and black, are all produced from the leaves of the evergreen species Camellia sinensis. Differences arising from the different degrees of fermentation during tea production. Black tea is commonly mixed with milk before it's consumed in countries including the United Kingdom and the United States. The researchers in the present study found that when black tea was directly applied to cells containing QCNQ5 channel, the addition of milk prevented the beneficial KCNQ5 activating effects of tea. However, according to Abbott, we don't believe this means one needs to avoid milk when drinking tea to the advantage of the beneficial properties of tea. We are confident the environment in the human stomach will separate the catechins from the proteins and the molecules in the milk. We otherwise block catechins' beneficial effects. Regardless of whether tea is consumed ice or hot, the temperature is achieved after the tea is drunk, as human body temperature is about 37 degrees Celsius, explained Abbott. Thus, simply by drinking tea, we can activate its beneficial antihypertensive properties. A study was supported by the National Institutes of Health, the National Institute of General Medical Sciences, the National Institute of Neurological Disorders and Stroke, and the Lundbeck Foundation in Denmark's Free Folkens Fund. So yeah, there's some, for most people, drinking organic green tea or black tea or white tea in moderation is fine. Some people with histamine issues might have problems, even though there are antihistamine properties within green tea. But it is fermented, and black tea is fermented, so people with histamine issues might react negatively to tea consumption. Tea also contains caffeine as well, and some people are sensitive to caffeine. Drink caffeine, it gives them the jitters. They can't sleep at night, they have insomnia, or they get anxiety. They might have to drink white tea, which has less caffeine, but would also have lower levels of these catechins, which are at their highest in green tea. And also people with oxalate issues may have problems with green tea and black tea, maybe because it does contain oxalates, even though the soluble oxalate amount, depending on certain brands of tea, of green tea or black tea, may be lower than others, so it just depends. But overall, the occasional ingestion of tea can be beneficial, unlike, drinking energy drink in the morning. Study finds 2 servings of fish per week can help prevent recurring heart disease. Analysis: several large studies involving participants from more than 60 countries, spearheaded by researchers from McMaster University, has found that eating oily fish regularly can help prevent cardiovascular disease in high-risk individuals, such as those who already have heart disease or stroke. The critical ingredient is omega-3 fatty acids, which researchers found was associated with a lower risk of major cardiovascular disease events, such as heart attacks and strokes, by about a sixth in high-risk people who ate 2 servings of fish rich in omega-3 each week. There is a significant protective benefit of fish consumption of people with cardiovascular disease, said lead co-author Andrew Minty, associate professor of research methods, evidence, and impact at McMaster, as a principal investigator at the Population Health Research Institute. No benefit was observed with consumption of fish in those with heart disease or stroke. The study has important implications for the guidelines of fish intake globally, and indicates that increasing fish consumption of particularly oil fish in vascular patients may produce a modest cardiovascular benefit. Minty said people at low risk for cardiovascular disease can still enjoy modest protection from cardiovascular disease by eating rich fish and omega-3, but the health benefits were less pronounced than those high-risk individuals. The study was published in JAMA Internal Medicine on March 8th. The findings were based on data from nearly 192,000 people in 4 studies, including 552,000 with chronic cardiovascular disease, and the only study conducted on all 5 continents. Previous studies focused mainly on North America, Europe, China, and Japan, with little information from other regions. This is by far the most diverse study of fish intake and health outcomes in the world, and the only one with sufficient numbers with representation from high, middle, and low-income countries from all inhabited continents of the world, says study co-lead Dr. Salim Youssef, professor of medicine at Michael D. Groot School of Medicine and executive director of the PHRI. So yeah, I have a blog vastly covering omega-3 fatty acids on Fix Your Gut. Of course, wild-caught fish is always best, and you want to stay away from high-mercury fish if all possible. So you want to ingest, low-mercury tuna rarely. Some brands that are good for low-mercury tuna are like Wild Planet, American Tuna. You want to get albacore tuna, of course, or Safe Catch tuna. Those are 3 low-mercury brands. And you want to ingest low-mercury seafood like wild-caught salmon, wild-caught cod, wild-caught flounder, wild-caught shrimp, wild-caught lobster, wild-caught crab, wild-caught sardines. And there's many different brands of fish oil, but you want the ones that are less oxalated and oxidized and are in the triglyceride form, and I have those listed brands on Fix Your Gut. And covering hidden forever chemicals. Research from the Harvard John A. Paulson School of Engineering and Applied Sciences Found large quantities of previously undetected compounds from the family of chemicals known as PFAS in 6 watersheds on Cape Cod using a new method to quantify and identify PFAS compounds. Exposure to some PFAS, widely used for their ability to repel heat, water, and oil, are linked to a range of health risks, including cancer, immune suppression, diabetes, and low infant birth rate. The new testing method revealed large quantities of previously undetected PFAS from fire retardant foams and other unknown sources. Total concentrations of PFAS present in these watersheds were above state maximum contaminant levels, MCLs, for drinking water safety. We developed a method to fully capture and characterize PFAS. We should develop a measure to fully capture and characterize all PFAS from fire retardant foams, which are a major source of PFAS to downstream drinking water and ecosystems. But we also found large amounts of unidentified PFAS that could have originated from these foams, said Bridger Rule, a graduate student of SEAS and first author of the study. Traditional testing methods are completely missing these unknown PFASs. The research will be published in Environmental Science and Technology, PFASs. PFAS, per- and polyfluoroalkyl substances, are present in products ranging from fire retardant foams to nonstick pans. Nicknamed forever chemicals, due to their long lifespan, PFAS have built up in the environment since they were first used in the 1950s. Despite the associated health risks, there are no legal enforceable limits for PFAS chemicals in drinking water. The EPA's provisional health guidelines for public water supplies only cover PFOS and PFOA, 2 common types of PFAS. Massachusetts, along with a few other states, have gone further by including 6 PFAS in their new MCLs in drinking water. But there are thousands of PFAS chemical structures known to exist, several hundred of which have already been detected in the environment. We are simply not testing for most PFAS compounds. So we have no idea what our total exposure is to these chemicals, and health data associated with such exposures are still lacking, said Elsie Sunderland, the Gordon McKay Professor of Environmental Chemistry at SEAS and senior author of the paper. The standard testing methods used by the Environmental Protection Agency and the state regulatory agencies only test for 25 or fewer known compounds. The problem is overwhelming majority of PFAS compounds are proprietary, and regulatory agencies can't find what they know exists. The new method developed by Sunderland and her team can overcome that barrier and account for all PFAS in a sample, CSI PFAS. That's funny. CSI PFAS. PFAS are made by combining carbons and fluorine atoms to form one of the strongest bonds in organic chemistry. Fluorine is one of the most abundant elements on Earth, but naturally occurring organic fluorine is extremely rare, produced only by a few poisonous plants in the Amazon and Australia. Therefore, any amount of organic fluorine detected in the environment is sure to be human-made. PFAS compounds found in the environment come in 2 forms, a precursor form and a terminal form. Yeah, that fluorine is going to have issues, cause issues with the functioning of the thyroid gland as a halogen and interfere with iodine intake. Most of the monitored PFAS compounds, including PFOS and PFOA, are terminal compounds, meaning they will not degrade under normal environmental conditions. But precursor compounds, which often make up the majority of PFAS chemicals in a sample, can be transformed through biological and environmental processes into terminal forms. So while the EPA or state agencies may monitor PFAS concentrations, they're still not detecting much of a huge pool of PFAS precursors. It's a problem. This is where the new method comes in. The research first measures the organofluorine in a sample. Then, using another technique, they oxidize the precursors in that sample and transform them into the terminal forms, which they can measure. From there, our team developed a method of statistical analysis to reconstruct the original precursors, fingerprint their manufacturer origin, and measure their concentration within the sample. We're essentially doing chemical forensics, said Sunderland. Using this method, Sunderland and her team tested 6 watersheds on Cape Cod as part of a collaboration with the United States Geological Survey, a research center funded by National Institutes of Health and led by the University of Rhode Island that focuses on the sources, transport, exposure, and effects of PFAS. The team focused on identifying PFAS from the use of fire retardant foams. These foams, which are used extensively at military bases, civilian airports, and local fire departments, are a major source of PFAS and have contaminated hundreds of public water supplies across the United States. The research team applied their forensic methods to samples collected between August 2017 and July 2019 from the Childs, Quashnet, Mill Creek, Marston Mills, Mashpee, and Santuit watersheds on Cape Cod. During the collection process, the team members had to be careful what they wore since waterproof gear is treated with PFAS. The team ended up with decades-old waters to prevent contamination. The sampling sites in the Childs, Quashnet, and Mill Creek watersheds are downstream from a source of PFAS from fire-retardant foams. The Quashnet and Childs from the Joint Base Cape Cod Military Facility, and Mill Creek from Barnstable Fire Training Academy. Current tests can only identify about 50% of PFAS from historical foams, products that were discontinued in 2001 due to high levels of PFOS and PFOA, and less than 1% of PFAS from modern foams. Using their new methods, Sunderland and her team were able to identify 100% of all PFAS compounds and the type of fire retardant foams that were used for decades at Joint Base Cape Cod and Barnstable County Fire Training Academy. Our testing method was able to find these missing compounds that have been used by the chemical industry for more than 40 years, said Sunderland. Wow. That's a lot of environmental contamination. The tests also revealed high quantities of PFAS from unknown sources. Our counting of PFAS from firefighting foams could not explain 37 to 77% of the organic organofluorine that was measured, said Rule. There's huge ramifications for not only our understanding of human exposure, but as far as much from PFAS is discharging into the ocean and accumulating in marine life. To follow up these findings, Rule is currently working with NIH to identify some of the health impacts of PFAS for contemporary firefighting foams using toxicological studies. Sunderland's team is continuing to study unknown PFAS to better identify their sources of potential accumulation in abundant marine food webs on Cape Cod. That was an interesting article. A new review of existing evidence proposes 8 hallmarks of environmental exposures that chart the biological pathways through which pollutants contribute to disease: oxidative stress and inflammation, genetic alterations and mutations, epigenetic alterations, mitochondrial dysfunction, endocrine disruption, altered intercellular communication, altered microbiome communities, and impaired nervous system function. So this is how toxins and pollutants make us sick. They cause oxidative stress in our cells throughout our body, inflammation throughout our cells in our body, genomic alterations and mutations. Okay, so they can actually alter and mutate our own genome. Okay, that's really interesting. Epigenetic alterations, which would mean how genes are expressed. Okay. Mitochondrial dysfunction, which would harm our mitochondria. Endocrine disruption, which would harm our ability to produce and utilize hormones. Altered intercellular communication, communication of cells. Altered microbiome community, so it would change our microbiome, and impaired nervous system function. This study by the researchers at Columbia University Mailman School of Public Health, Ludwig Maximilian University, and Hasselt University is published in the journal Cell. Every day we learn more about how exposure to pollutants in air, water, soil, and food is harmful to human health, says senior author Andrea Baccarelli, MD, PhD, Chair of Environmental Health Services at Columbia Mailman School. Thus understood, however, specific biological pathways through which these chemicals inflict damage on our bodies. In this paper, we provide a framework to understand why complex mixtures of environmental exposures bring about serious illness even at the relatively modest concentrations. We are continually exposed to a mixture of pollutants which lead to changes in our bodies in multiple domains, from conception to old age. They govern gene expression, train and shape our immune systems, trigger physiological responses, and determine well-being and disease. Paper summarizes evidence of 8 hallmarks of environmental insults. Oxidative stress and inflammation. When antioxidant defenses are depleted, inflammation, cell death, and organ damage occur. Genomic alterations and mutations. An accumulation of DNA errors can trigger cancer and other chronic diseases. Epigenetic alterations. Epigenetic changes after the synthesis of protein responsible for childhood development and regular function of the body. Mitochondrial dysfunction. A breakdown in the cellular power plant may interfere with human development and contribute to chronic disease. Endocrine disruption. Chemicals found in our environment, food, and consumer products disrupt the regulation of hormones and contribute to disease. Altered intracellular communication. Signaling receptors and other means by which cells communicate with each other, including neurotransmission, are affected. That's interesting. Altered microbiome communities. An imbalance in the population of bacteria and other microorganisms in our body can make us susceptible to allergies and infections. Impaired nervous system function. Microscopic particles in air pollution reach the brain through the olfactory nerve and can interfere with cognition. Not all environmental exposures are harmful. The researchers note that exposure to nature has been reported to have beneficial impacts on mental health, of course. These 8 hallmarks are by no means comprehensive and do not capture the full complexity of the chemical and physical processes of environmental exposures, including mixtures of exposures over the short and long term. Further research is needed to understand the complex mechanisms by which exposures affect human biology and how altered processes interact to contribute to disease or confer health benefits across the life course. Oh, thank you, Howdy Stranger. Thank you for the kind word. We need research to expand our knowledge or disease mechanisms going beyond genetics. Advances in biomedical technologies and data science will allow us to delineate the complex interplay of environmental insults due to the single-cell level. This is Beccarelli. The knowledge will help us develop ways to prevent and treat illness with serious environmental changes like air pollution and climate change. Yeah, they got to add that in. Most of all, we need strong local, national, and intergovernmental policies to ensure healthy environments. So yeah, pollution's a problem. Air pollution, we just read about water pollution with fluorine compounds. And yes, this is what the pollution will do to your body. These are 8 verified ways that pollutants will make you ill. Now, there's dosage makes the poison, and not every pollutant is the same, right? And some are more toxic than others and less. And many of them, will cause different ones, through this list, 1 through 8. They might not cause all of them. They might cause only a few. They might cause all of them. It depends on what you're dealing with. Arsenic would would do 1 through 8. But it just depends. There's chronic exposure, there's acute exposure. But it's interesting they broke it down into that, made that list. And the list is right. Those are ways that it does harm you. After a concussion, no more sitting in the dark. Concussion, a form of traumatic brain injury, is a common injury among children and teens. Concussions can have adverse effects on physical, cognitive, emotional, and sleep health. Clinical guidelines for managing concussion in children and teens traditionally recommend complete physical and cognitive rest until symptom resolution, followed by a gradual return to activities like school and sports. Yeah, that's true. They used to tell you not to sleep when you have a concussion, but as long as you don't pass out, it's safe to take a nappy nap. The brain remodels and heals itself when you're sleeping. These guidelines are often disputed and based on expert consensus and opposed to strong evidence. The challenge has been how to quantify the amount of physical and cognitive activity that children and teens should engage in during recovery. A new study by a researcher at the Center for Injury Research and Policy, Sports Medicine, and Emergency Medicine Nationwide Children's Hospital investigated objectively measured self-paced physical and cognitive activity across the first week of post-concussion. The study published today in the Journal of Head Trauma Rehabilitation found that self-paced physical and cognitive activity during the first week after sustaining a concussion, although neither hastened nor prolonged concussion recovery in children and teens. These findings indicate that children and teens with concussions may have some flexibility to determine their own activity levels during recovery. Physicians may encourage safe, non-contact, light physical activity, like walking would probably be an example of that, which, walking in the sun, sun shining, you're setting your circadian rhythm. Depending on how close you are to the equator, it's still the winter. You're entering spring. But yeah, I guess we'll see. But yeah, you'll be able to make vitamin D, which would help modulate your immune system. The UV index is greater than 3. The light entering your retina would help produce serotonin and norepinephrine, and help, later if your methylation cycles are good, methylate into melatonin at night through all your cells of your body, through the mitochondria. So, walking, I wouldn't see why walking would be detrimental for someone with a concussion. Physicians may encourage safe, okay with that. If it does not make use of concussion symptoms worse, yeah, if you're overexerting yourself, that could be a problem. Of course, I'll get you jacked up. Also, can you emphasize appropriate amounts of necessary rest? Yeah. Studies participants were 11 to 17 years old, seen within 72 hours of injury in the emergency department of a concussion clinic through Nationwide Children's Hospital, received a physician-confirmed diagnosis of concussion. They wore 2 devices, one to measure the physical activity and sleep around the clock. Yes, sleep, one of the most important things. And quality of sleep, too. You want to sleep in darkness. And if you can, turn off as many non-native EMF producing devices that you can in your house, So turn off your router, put your phone on airplane mode if you can. Not everyone can. At least put your phone away from your bed, maybe on your dresser. Turn off your game systems, turn off your computers. Simple stuff. and one to measure the cognitive activity outside of school. Participants rated their daily concussion symptoms by completing the post-concussion symptom scale during the first week post-injury. Researchers found that while daily physical and cognitive activity increased across the first week post-injury, daily post-concussion symptoms decreased. Increased daily step count was associated with increased likelihood of early symptom resolution. However, this association was not statistically significant after adjusting for acute post-concussion symptoms and other covariates. Studies that first objectively measure self-paced cognitive activity during the first week post-injury, said Ginger Yang, PhD, MPH, lead author of the study and principal investigator in the Center for Injury Research and Policy at Nationwide Children's. While increased physical and cognitive activity may help reduce post-concussion symptoms, reduced symptoms may also lead to increased physical and cognitive activity levels, highlighting the need for further research to better understand this bidirectional relationship. Such research will provide evidence of when an individual is ready for physical and cognitive activity after concussion and what level of activity is most appropriate. The information could be used by clinicians to inform treatment decisions, including individual physical and cognitive activity recommendations post-concussion. Concussions behave somewhat like snowflakes in that they're all not the same, and the specific effects of concussion on young patients varies greatly. One size of treatment does not necessarily fit all, says Thomas Pomeroy, DO, co-author of the study and division chief of sports medicine at Nationwide Children. Yeah, I mean, you're right, Thomas. The study of this, everyone's different. We have different microbiomes, we have different genetics, we have different epigenetical expression, we have different nutrient storages in our bodies, different immune functioning. Straight to the studies, it appears that children and adolescents with concussions are generally pretty good at self-pacing their physical and cognitive activity, so as not to prolong their recovery. With the guidance of physicians, that may be room for personalized treatment plans based on symptom burden, while still following important guidelines protecting the patient from repeat head injury during the recovery time. All right. Invasive weed may help treat some human diseases, research found. Native to the southeastern United States, a weedy grass has spread northward to Canada and also made its way to Australia and Japan. Andropogon virginicus grows densely packed and up to 7 feet tall, disrupting growth patterns of other plants and competing for resources. When burned, it grows back stronger. There's no way to effectively remove the weed once it's invaded, but there might be a way to use it to human advantage. It sounds kind of like kudzu. An international team of researchers found that A. virginicus extracts appeared to be effective against several human diseases, including diabetes and cancer. The results were published on December 31st, 2020 in the special issues of Plants, titled Biological Activities of Plant Extracts. If I was a researcher, I could be a plant extract researcher. I think that'd be pretty cool. And I think that, if I was a researcher, that might be one thing I would do, is I would, study plants. I think that'd be cool. A. virginicus is an invasive weed that seriously threatens agricultural production and economics worldwide, said paper author Tran Dang Xion, associate professor in transdisciplinary science and engineering program in a graduate school of advanced science and engineering at Hiroshima University. It's in Japan. Yeah, the Japanese would give a title like that long of a title. However, no solution efficiently utilizing and tackling this plant has been found yet. In this paper, we highlight the potential application of A. virginicus extracts in the future of medical production and therapeutics of chronic diseases, such as type 2 diabetes and blood cancer, which can deal with both the crop protection and human health concerns. Researchers found high levels of flavonoids in the samples they extracted from the weed. Yeah, you'll find that a lot of research coming out of Japan is scientific research is kind of advanced in a lot of ways, some even compared to researchers coming out of the United States. These plant chemicals have significant antioxidant and anti-inflammatory properties, according to Xion. When tested against a variety of cell lines, extracted plant chemicals bonded to free radicals, prevented damage to the cells and skin levels. This helped prevent age spots by inhibiting a protein called tyrosinase. Among other deepful health actions, this bonding also helps prevent knock-on cellular actions that can lead to type 2 diabetes. The team also specifically applied the extracted chemicals to a line of chronic myelogenous leukemia, rare blood cancer. Extract appeared to kill off the cancer cells in vitro. Swan said that researchers plan to establish a comprehensive process to isolate and to purify the compounds responsible for known biological properties, as well as to identify new uses. They will further test the therapeutic agent effects of the compounds with the eventual goal of preparing functional pharmaceuticals for human use. Although A. virginicus has been considered a harmful invasive species without economic value, its extracts are promising sources of antioxidant, anti-diabetic, anti-tyrosinase, and anti-tumor agents, so on said. And finally, the gut microbiome influences the metabolism of processed foods. Studies of the microbiome in the human gut focus mainly on bacteria. Other microbes that are also present in the gut, viruses, protists, archaea, and fungi, have largely been overlooked. New research in mice now points to a significant role for fungi in attested the communities of molds and yeast known as the mycobiome. You got the microbiome, you got the mycobiome. Their active interface between the host and their diet. We showed the gut microbiome of healthy mice was shaped by the environment, including diet, that was significantly correlated with the metabolic outcomes. Said Kent Willis, medical doctor and assistant professor at the University of Alabama at Birmingham, and co-corresponding author of the study published in the journal Communications Biology. Results support a role for the gut microbiome in the host metabolic adaptation, and these results have important implications regarding the design of microbiome studies and the reproducibility of experimental studies of the host metabolism. Willis and colleagues looked at fungi in the gingium, which is the second part of the small intestine, of the mouse small intestine, site of the most diverse fungal population in the mouse gut. So you got the duodenum, you have the gingium, and you have the ileum. Then you have the cecum, which is the first part of the large intestine. You have the ileocecal valve that separates the ileum and the cecum. They found the exposure to processed diet, which is representative of a typical Western diet rich in purified carbohydrates, led to persistent differences in fungal communities that significantly associated with a different deposition of body mass in male mice compared to mice found in standardized diet. The researchers found that the fat deposition in the liver, transcriptional adaptation of metabolically active tissues, and serum metabolic biomarkers were all linked with alterations in fungal community diversity and composition. Variations of fungi from two genera, Theromyces and Saccharomyces, were the most associated with metabolic disturbance and weight gain. Elevated Saccharomyces in the gut can actually cause a condition called autobrewery syndrome. When you ingest mostly any type of mono or disaccharide, you actually start producing alcohol in your gastrointestinal tract, and if enough is produced, people actually get drunk from even small amounts of sugar consumption. It's crazy. The study has had an ingenious starting point. The researchers obtained genetically identical mouse from 4 different research animal vectors. It's known that gut bacterial communities vary markedly by vendor. Similarly, the researchers found dramatically different variability by vendor for the gingium microbiomes, as measured by sequencing internal transcribed spacer RNA, rRNA. Wow, so they are actually measuring the live instead of measuring DNA through PCR. They're measuring live microbiomes. Okay, that's good testing. Good job, lab. Good job doing the right stuff. A baseline mice from one of the vendors had 5 unique fungal genera, and mice from the other 3 vendors had 3, 2 and 1 genera, respectively. They also looked at inter-kingdom community composition, meaning bacteria as well as fungi, and found large baseline bacterial community differences. From the initial fungal and bacterial diversity, then they measure the effects of time and differences in diet, standardized chow versus the highly processed diet, on fungal and bacterial community composition. The researchers also addressed the fundamental question: are the fungal organisms detected by the next generation sequencing coming from the diet? Are they true commensal organisms that colonize or replicate in the gut? Well, that's a good question. Is it the fungi on the food they're eating? Because there is some contamination in the food that we eat. Or is it directly coming from their gut? Now, the RNA should show that. That's a good question. Whoever wrote this write-up and these researchers, they did a real good job. They compared sequencing of the food pellets, which contain some fungi, exactly, and the contents of the mouse gingium to show the gingium fungi were true commensal colonizers. Good job. This is a good study. The study led by Willis and co-corresponding author Joseph Pierre, Ph.D., and co-first authors Talia S. Mims and Quizi Al Abdullah, Ph.D., from the University of Tennessee Health Center, Memphis, Tennessee, showed the variations in the relative abundance and composition of the gut microbiome correlate with key features of host metabolism. This lays the foundation towards understanding the complex interkingdom interactions between bacteria and fungi, and how they both collectively shape and potentially contribute to host gut homeostasis. Our results highlight the potential importance of the gut microbiome on health and have the implications for human experimental metabolic studies, Pierre said. The application for human microbiome studies, which often examine only bacteria and sample only fecal communities, is that the microbiome may have unappreciated effects on microbiome-associated outcomes. So that's really interesting. All right, everybody. That's it for today's stream for Fix Your Gut News and Views, week of March the 11th, 2021. Take care. Hope you have a good day.